welcome to the Bioinformatics chat. This year, after a long break, we had another installment of the Bioinformatics contest. And today we'll be discussing the final round problems from the contest. And to do that, I'm joined by Matt Holt. Welcome, Matt.
Hi.
And Maksym Kovalchuk. Welcome, Maksym.
Hi.
Oh, and I should introduce you, right? So Matt is the second prize winner and Max is the first prize winner this year. So congrats on the great performance. And I'm guessing that a lot of people are wondering how they can develop, how they can learn to solve these types of problems, and how you guys came to be able to do this. Could you talk about your background and what kind of training do you think allowed you to perform so well this year? And Matt, let's start with you.
Yeah, so like you said, I'm Matt Holt. My, I guess, road to this point in my life is I went to both undergrad and grad school in computer science. And it wasn't really until grad school that I started to get into more genetics-related things. So my advisor at the time was studying mouse genetics, so I kind of started to learn how to apply the skills I had learned up to that point to the field of genetics. More recently, I am now working at a clinical lab in Huntsville, Alabama. And since we're a clinical lab, of course, we're doing human genetics. So pretty much everything I've been doing for the past 5-ish years has been focused on rare disease in humans. So a lot of the skills are, of course, computer science related and algorithms and all those kind of fun things. But it's just the place I'm applying them right now is human genetics. So that's kind of the short answer of how I got to where I am.
Cool. And do you find that your computer science education helps you in your day-to-day job, or is it just for the contest?
Yeah. Clearly, there's a lot of really hard problems in genetics. Part of that's just the fact that the human genome is huge, right? It's like 3 billion base pairs. And the way we gather data to do any sort of analysis on that requires really efficient methods to do any sort of analysis, just because the data is so big. So, I'm obviously not tapping into deep wells of algorithmic knowledge every day, but, when I get to it, it's really fun and certainly comes up fairly frequently.
Cool. And Max, what about you?
I started programming in the school and continue it in the university. So now I'm third-year student in computer science. Basically, I do a lot of competitive programming, but from last 2 years, I switched into more optimizational problems. Those are more like when you doesn't have exact solution, which is typical for bioinformatics and some other fields. Yeah, and I find these problems more enjoyable because in competitive programming you usually have 5 hours or less. For these problems you sometimes have week, sometimes have day, sometimes you can have several years in case it's some scientific project. And it's way more interesting for me.
And do you have a general interest in bioinformatics, or is it just for the sake of contest?
I think it both. So basically, I am very competitive. So if I see some possibility to participate in contests, so it's probably 100% that I will participate, even if I have exams or something. And but nonetheless, about a year ago, I saw some courses on Coursera about bioinformatics, and I take them just for interest.
Cool. And one other question I want to ask you guys before we move to discussing the problems themselves is So the way, for our listeners who don't know, the way the contest is structured is that there are two rounds. The qualification round, which we won't be discussing today. This year, I think it was pretty easy. And that lasted for 1 week. And then there is the final round, which lasts for one full day, for 24 hours. And so every time when I participate, I struggle with this decision, how to allocate time, how much sleep do I get, right? So there is the tension between getting a good night's sleep and being rested the next day and tackling more problems versus just getting more raw time to do stuff and get very little sleep. So I'm curious how you approach this.
Yeah, so for me at least, the contest starts this year. It started at 8 a.m. my time. So I basically had a full day, and then it was towards the end of the contest where, all the energy's drained for me and I just want to go to sleep. I think, let's see, I got a good 30-minute power nap right in the middle of that, about 3 a.m. for me. While I was waiting for something to run, I just said, all right, I'm going to go to sleep while this runs. And, the time management, it's a funny aspect because, I don't sleep much during the contest. I had that one nap and that was it. But the rest of it's just kind of trying to figure out which problem you think you can get the most points on for your effort. Sometimes, honestly, I'll go look at the scoreboard to see who's finished out certain problems. And if I see a bunch of people have finished problem 4, it's like, okay, I can do that one. I'm going to go see what it is and check it out. So there's kind of a weird, almost game theory aspect to all of this, where you're looking at how well others are doing in certain areas and trying to see if that translates into something you can do as well.
Yeah, for sure. Although maybe this year it didn't matter as much because at least both of you, I think you solved all of the problems. Maybe not for the full score, but pretty much you did everything. So maybe it didn't matter that much in which order.
Yeah, there was only, I think there was the one problem that had a kind of open-ended scoring system, but all the other ones seemed to have an exact answer. It was just seeing if you could find it.
Did you stay up until 8 a.m. next day?
I did. And then they had the actual, they had like a live stream going on. So I stayed up a little bit longer just to say hey to the live stream. And then I passed out for a few hours after that. I don't know if you had a similar experience, Max, but that was basically what I did.
Yes, well, as for me, first about order of the problems, I think there is one trick that if you start with harder problem and you doesn't solve it at the beginning, while you're solving other problems, you somewhere inside your mind solve the first problem, which is the most hard. So I prefer to at least read and spend at least 15 minutes for all problems, and then we'll see which one I should work. Because usually to solve the problem for the full score or for the max feasible score, I test 2 or 3 different solutions. And if you test the first solution near the end of the round, it's you probably wouldn't have time to come up with second and third one. Yeah, and about the time management, so I basically, while I'm a student, I don't have all that much active life, and I can allocate several days for the contest. So I sleep a lot before the contest starts, and I can do anything. I don't do anything after contest ends because I know that I will be tired a lot. And about sleeping during the contest, actually I plan to not sleep at all, but somewhere at the middle I felt that I just can't. I almost dying, but so I sleep for 1 hour, but that's all. Maybe also half an hour for eat some food or something, but no. everything, every possible second, because I remember the last submission was half an hour before the end of the contest, so it really matters for me.
I am jealous of the ability to get that sleep beforehand. I got kids at home, and that doesn't quite pan out for me.
Yeah.
All right. So now let's get into the problems. And the first problem is genotype imputation. And that's where you get, so as your input, you get binary strings, right? So you have basically a set of SNPs, single nucleotide polymorphisms, which have just two alleles. And so you get the population for which you know the genotypes. And so for those individuals, you get two binary strings, which are the two haplotypes for that individual. And then you have a bunch of unknown or partially known genotypes where first you have just one binary string, and some of the binary digits are replaced with question marks, which means you don't know the allele at that locus. And your goal is to guess correctly the unknown alleles. And one thing I didn't mention is that when you get partially known genotypes, they're no longer binary strings, right? They're sort of ternary strings, because you get the count of alleles, which could be 0, 1, or 2. And so, yeah, the naive approach is just to look for the known genotypes which are most similar to the partially known ones, right? But then the question is how large of a context you take into account, right? So if you have a question mark in the partially known genotype, you can take a window of like 1 nucleotide or 2 or 3 and look for known genotypes that are most similar in that window.
So yeah, I initially did something very similar to that naive approach that you were talking about. I think I even took it one step further back, which was I just picked the 2 haplotype combinations that got me closest to the observations, and just said, all right, whatever that adds up to is the answer, which is not very good because it doesn't factor into lots of things like recombination and just variation within populations and things like that. But that was kind of my kind of first pass at it, as Max was talking about the different versions of things. But the problem itself, it's very well studied, I think, in bioinformatics. There's a lot of reasons people want to impute things, whether that's just we have an array that's, it's not very, it doesn't have a lot of variance on it. So you want to kind of figure out what might be in between, or, any sort of low resolution test like that. Imputation becomes critical. As for your question of how I ultimately did it, I actually took a kind of machine learning approach to this one. This was I didn't actually think it would work, but it kind of panned out pretty well, to be honest. Where I basically just started combining those haplotypes into kind of a training set, if you will. And then once I had that training set built up, I would just try to predict whether it was a 0, 1, or 2 for all the ones that were missing. And yeah, the window definitely mattered. I think I initially only used like 5 observations around, but as I increased that, the score went up. Whether the correctness went up or not, I'm not sure, but the score at least went up. So I'm assuming the correctness did as well. And yeah, I don't remember where I ended up at. I think I got up to at least 63-ish observations around, but that varies heavily from problem set to problem set because some of them had more observations than others. So that's kind of how I went about it.
So when you say you combined these into a training set, but what was your model like? How did you actually went from the training set to the predictions?
Yeah, I mean, the models I just used like easy ones that you can implement in Python from sklearn. So random forest was in there. I eventually switched to gradient boosting because it got me better answers. It was a bit slower, but it was better in terms of the actual results.
Oh, that is so cool. It never actually occurred to me to use a machine learning library.
It's one of those, it's one of those weird problems that I don't think machine learning is a typical solution here, but I didn't know how to implement an imputation package from scratch or even how to use the ones that are available just because I haven't done it before. So I basically said, how would I take a situation where I'm trying to predict something? Well, that's kind of what machine learning does, is it makes predictions. So that's kind of how I got to that solution.
I should remember this idea for the next contest. And by the way, we should mention, I'm looking at the scoreboard right now, and I'll give a bit of background. So the problem number 1 is one of those sort of harder problems where no one and for no problem made a full score. And I think not even close.
Max got the closest, right, for that one?
Except one test case. I think on one test case you have better score.
Yeah, I think Max had the high score, and then I had the second overall, and then it kind of fell out from there.
My point is that the highest score, so for example, if the maximum is 150 points, then for example, for the very first test case, the maximum is like 116 out of 150, or for the third subproblem, it's 181 out of 250. So it's not even like 10% difference from the full score, but it's there's a lot of room for improvement, let's say. And so, did you guys think that this was the hardest one?
I mean, in terms of scoring, it clearly was, because no one got close to getting the maximum points. In terms of whether it's the hardest problem period, I'm not sure. It's kind of hard to say.
I think it's the most memorable for me, but if the most hard, I don't think.
Is this the one you were working on late in the night, so to speak? This was the last one I was working on.
Yes, it's last one.
Well, was that also the one where the two of you were competing the hardest on? Like that pretty much determined the outcome? Because you had a very close, right? You have very close scores.
I know that's the one I was working on at the end of the night, because I thought I could squeeze more points out of it.
Yeah, there's a lot of potential there, right? In terms of the points.
Yes.
So Max, what was your approach, and what were your thoughts?
So basically, I have like two approaches that were not good enough. Like 3 hours before the end of the contest, I done all 4 other problems and come back to this one. And it was hard to come up with something, so I have like one stupid idea that worked out in the end. But so first, we have like n pairs, and I made like n square pairs. So this like simply increases the amount of data, and because it's like combining different haplotypes, it's like you have n parents and you have n square child, so it should be okay. And the second, the prediction. So basically, I predict based on one site. So you talk about that you take some window, maybe it's like 63 sites, but I use only one, and I brute force all the known sites, and I choose one that gives the best prediction on the test case, on the test data, so this n square haplotypes. And just choose one. And when you have like one predictor, you have 3 possible values for the predictor and for the one that you predict, and this makes like 3x3 matrix, and then just choose maximum value from each line or from each column, and that basically is a prediction. So for the particular site.
Right. So for every site, you look for another single site which will work as a predictor.
Yes.
Yeah, that is also like a very cool idea that didn't occur to me.
I'm a little surprised that worked. That's cool.
Yes, I'm also very surprised because it shouldn't work because it's like only one site. How can it work? But I don't know. I haven't other options in the end, so I just implemented it.
Yeah, but it's in the spirit of machine learning, it's a very data oriented way, because the approaches I tried were just based on my a priori, thoughts, like what would be a good predictor, versus both of your solutions. They use this really as a training set.
Yes.
That's very cool. The next problem is called causative mutation. And so here you have a haploid version and a diploid version. So we'll start with the haploid version. In the haploid version, you get a bunch of haplotypes and a marker, whether it's a positive or negative for some trait. And your goal is to figure out which locus is responsible for that trait. And you don't need to determine it exactly. You can provide an interval within which you believe the locus lies. If you are correct, then you get a number of points inversely proportional to the size of the interval. So the larger interval you give, the more uncertain your prediction is, the fewer points you get. But if you are wrong, if the causal locus is outside of the interval, you get 0, of course. What were your approaches here?
So basically, this problem has limited number of attempts because like you can try to send intervals equal to of size 1 for each test case, and like send a lot of these attempts and get full score without solving the problem. But the interesting thing is that you can base the biggest, like the size of the length of the input data is about one maximum length is about 1,000, and you have 10 attempts for each test case. So basically, you can do binary search on this on the length, and without writing any code, just doesn't think and just do binary search, and that's all. But I also tried to a little bit optimize this, because it's also take a lot of time to every time update the intervals and send attempt again and wait for the scoring. So basically what I did, if we have like 2 strings and we know that one have a marker of some disease and other one doesn't, and they have some interval that is the same, that have all the same letters, A, C, G, T. This means that this interval probably doesn't have mutation for the disease, because like they are this interval is the same for both healthy and ill genome. But it's not exactly true. So first, I take only long intervals that are the same, because if it's like one site and the other letters that near this site are different, it's doesn't say anything because it's probabilities that two sides are the same 1/4. So I take only long enough intervals, and the second I make some thresholds. So there should be enough such intervals that cover some site. So if some site is covered by like 20 intervals, it's probably not doesn't have marker for the disease. And if like 0 or only 2 or 3 intervals cover some site, then probably it's the answer. But it turned out that it's not work very well on all test cases. So I can say that half of test cases it gives a very accurate solution, but for the other half test cases, I still use a lot of bin search.
Yeah, I remember that like I implemented some kind of informational theoretical solution. Like I calculated like a mutual information between the traits and the various alleles at the loci, and looked for the maximum one. And sometimes it worked, and sometimes I did have to resort to binary search. But I'm looking at the scoreboard now, and I don't have the full score for that problem. So I don't remember if I just was lazy enough to do binary search, or it didn't work. I don't know. And Matt, what were your thoughts?
Yeah, so this is, this problem, much like the last one, is a very classic bioinformatics problem. I have this set of patients who are sick, this set of controls who are not sick. Find the thing that is making the sick patient sick, right? And of course, depending on the resolution of your test, you again might not have the actual variant or change that's causing them to be sick, even in your test, which is where this kind of imprecision comes into this problem. But again, despite the fact that this is a very classic problem, this is not something I've particularly done before. So this is another area where I actually fell back on machine learning. This is the second one. This is the last one where I fell back on machine learning to do this. But I basically said, take another window, right? So take 5 of these observations we have and see how well those 5 will predict whether the patient is sick or not. And train the model, see how well it does, and you actually can plot it over the whole length of your genome, which is the sequence that they give you. And it turns out that that's really good at generating a peak, which is just, it's kind of what you would do in a GWAS type thing, right? Where you're doing this genome-wide association of which thing is causing my disease. So you can plot it, you see a peak, and I think actually for every answer, that got me to the ballpark. And then it was just a matter of shifting left or right a little bit to get the exact answer right, almost in like it was very binary in that way, kind of like what Max said, only instead of zoning in, I was zoning out a little bit because it was slightly to the right or left of where I actually had pinpointed from my initial solution.
Mm-hmm, cool.
Yeah, that part was a little unfun. The little manual twiddling of the result I got. I hope they don't do something like that again.
Well, it kind of was on you, right? That you didn't. Your solution actually, like, it sounds like no solution was good enough to produce the correct answer by its own.
I think it's also that reflects biology too, right?
Yeah.
A lot of people will do these GWAS studies and they'll get locus whatever matches my patient, but that's not usually the actual answer. It's something around there that is the thing they're looking for, and there's usually follow-up studies to figure out what exactly that is. So it was good in that it matched the kind of real world problems that we have, but it gave me an appreciation for the challenges around that problem as well.
Yeah, and in the real world, no one will tell you whether your interval is correct or whether you get the full score or not. You have to do experiments.
Yeah.
Oh, and there was also a deployed case. So was anything different in the deployed case?
For me, I just changed the inputs a little bit, and it was harder, right? There wasn't as clear of a peak for me.
They were more of bin search. Yeah.
It was just kind of like a scaled up version of the same thing to me anyway.
Okay, and the next problem number 3 is superspreaders.
I'm actually really curious about your solution here, Max.
Yeah, so you were given sort of like a graph in time, right? So you had several days, and for each day you were given the contacts that were to occur on that day and the probability of spreading the virus. And you had to find a person such that if the person is infected on day 0, that would lead to the most infected people on the given day T. And also, like, the scoring was in median, right? Not on average, but in median. So the simulation was done multiple times, and they would take the median number of infected people at the end. And so you had to provide this person who would infect the most other people in median. Yeah. So Max, how did you tackle this?
So basically, first, this problem have like this model with median, and also this model is kind of discrete because you either ill or not, and you have to make like 99 simulations, and that is very slow. So first, what I did is make it more kind of continuous. So I hold the probability that some person is ill. And then you have to do the simulation only one time. And this have this small problem that actually you have, in the end, you have the average number instead of median, but they are quite correlated. So if some person is super spreader, so it's in terms of median, it's probably the same in terms of average. Yes. And then when I have this better model, I simply test every person to be a super spreader. So it's kind of quadratic solution, and also a little bit of parallel programming, and that's all.
Yeah, so that's what I was curious about. It sounds like you had a brute force kind of solution once you have the model built, right?
Yes. And
yeah.
Also one thing, this problem also have limited number of attempts. But the thing is that when after I test every person, and if I choose one that have maximum average, I sometimes doesn't have full score. So I maybe sometimes try to take second or third to get full score.
Yeah, I mean, I did a similar thing here. I like you reduced it to just the probabilities and just summed up what the total likely infected was at the end of a simulation. But yeah, I could not think of a solution here that wasn't just kind of brute forcing it like that. I had a heuristic approach that allowed me to prune down which ones I actually did the full simulation on. But aside from that, it was still just run the simulation, get a number, and whichever one is the biggest is the one I gave to the scoreboard.
Well, I feel it can be pruned very well, but it's needs to work on this problem like for a week or something.
Yeah.
But not in a few hours when you're sleepy.
I will say this was my favorite problem, just because it seemed very timely given the state of the world.
Yes, and I also like this problem because pretty much all the problems are your given genome or something, and you have to do something with these letters or binary strings. And this one is like another type. It's more about like something a real world simulation and not about do something with genome.
I do wonder if there's some sort of non-brute force solution here. I mean, internet, tell us if there's a non-brute force solution.
Well, I always wonder how unfortunately this year we don't have anyone from the organizing team or the problem authors. But it's a big responsibility on the problem authors because you and I, we can just submit several attempts and see which one gets the highest score, but someone on that end has to actually figure out what solution should get the largest, the full score. And for some problems it's easier. So for example, for the causal mutation, you actually know what the causal mutation is because you have the raw data from which the problem was generated. But in this case, right? In this case, they have more time and more resources, so maybe they would be fine with the brute force approach. I don't know.
That would be my guess. Yeah.
By the way, I wasn't giving credit to the problem authors, so let's correct this. For the problem one, the author was Alexey Sergushichev, and the tests were done by Gennady Korotkevich. For problem two, causative mutation, the author was German Demidov, and the tests are by Nikolai Budin. And problem three is by Vitaly Aksionov, and the tests are by Alexandra Drozdova. All right. Anything else to say about Superspreaders?
Don't be one.
Perfect. And problem number 4 is minor haplotype, which is by Nikita Alexeyev. The tests are by Daniil Oreshnikov. Oh my god, the problem text is super long here, and I don't remember what it's about. So Matt, can I ask you to explain the problem?
Yeah, I'll take a stab at it. So if I remember correctly, this one was basically a situation where you get a bunch of sequencing of some sort of viral genome. And the question was, does this sequencing represent one viral population, or is there a smaller subpopulation within this one? Basically, has our virus mutated in some way is kind of the way to think of that. And there were a variety of parameters they gave you, like the sequencing error rate, a minimum number of variants for it to be a new strain, and I think like a minimum population level or something like that. And you're supposed to try to factor all of that into your problem to say, yes, this is just a single strain, or no, this is a second strain. And I think the second part of that was if there was a second strain, for some of the problems, they wanted you to tell them what that sequence was. And that was scored on some scale based on how close you were to the true second sequence. So that was the long and short of it, I think.
Yeah, that's a very good summary. And how did you approach it?
So this is the one I'm probably least satisfied with overall. So let me start with that. But the basic way I did this was I tried to take the sequences and cluster them into two clusters. So, you can take all these different sequences and say which ones are most like the other ones and form two cluster strings that you can then look at. And then the second step I would do is say, okay, are these two clusters significantly different based on whatever parameters they gave you for the problem? So if they weren't different, then of course I would say, okay, this is just one viral population, and that was the end of the problem. If they were different, I just did like a consensus of the viral sequence, the second population, and submitted that. But the hard part was figuring out whether it's one or two. That was by far the hardest part in my mind. And And so for me, I actually would take the group of subproblems for one of those problems and kind of I had this scoring system that I don't remember how I did it off the top of my head, but I could plot that and get kind of a linear degradation. And there would be one group that was very well clustered that was probably the ones that were just one viral population. And there was another group that was probably two. And then there were some in the middle where I wasn't exactly sure. And I think part of that's just because I didn't fully factor in all of the sequencing statistics and all these other numbers they gave you for the problem. I think if I had done that, I could probably have gotten a more precise solution overall. So this one was the one I was least satisfied with, but I think with more time, I could have done a better job on this one.
Cool. Max, what about you?
Yeah, so as Matt told, in some cases you may not have minor haplotype. In some tests, you have output only like one or two, which of these two cases. And think again for test where you can get full score without writing anything, because you just can try it for each test case one or two, and that's all. Yes, but actually it's the three other test case where you have to output actual minor haplotype first. One of them have this important feature that sequencing error is very low. So you have 200 bases, and about 2 bases are errored, and you have the difference between major and minor haplotypes is about 16 bases or more. So if you just compare major haplotype with something that you have in input, if difference is more than 8, then it's probably minor haplotype, else it's not. So this test case is very easy. Just count the number of difference between two strings. Yes, and for the other two test case, I again have some troubles in the beginning, and my last approach was something next. So I wanted to find which bases are different in minor haplotype. So basically what I did, I brute force all triplets, and if in some triplet I have the same value of the basis is the same, but different from major haplotype, probably this base is mutated and it's from minor haplotype. So after I take all triplets, I'll have some statistic in how many triplets this base is kind of special, and I just put some threshold. If it's special in more than, let's say, 20 triplets, then it's probably from minor haplotype. And after that, I don't actually remember how I choose haplotypes, but it's then easy if you have bases that are mutated.
I was going to ask Max, what did you actually code most of your stuff in?
C++.
Okay.
What about you?
Yeah, I was using Python, which is handicap in some way because it's slower, but also easier to make things just work sometimes.
Well, basically for me it's opposite. Probably I know that all the people say that it's easier, but probably because I'm all the time coding C++, it's easier for me, and it's probably the main language in competitive programming.
Yeah.
How easy or hard is it in C++ or in Python for that matter? Also curious to organize parallel computation, because and I think you mentioned, and from my own experience, I know that helps quite a bit if you can parallelize your code easily.
So at least in Python, and I think in this competition too, you can, if you can separate your problems into very clean subproblems, it's very easy to parallelize it. which in this contest, a lot of them are just, there might be 100 subproblems or something. And so it's very easy to run one problem at a time on different processors.
Multiple copies of the same script, but different processes.
Of the same function. Yeah, you can use multiprocessing pools and just say, run, this particular function. Here's an array of 100 inputs. Go do it 100 times, and it will just kind of take care of all that for you. So that part's a little easy. It's just a matter of, can you actually create those subproblems that are kind of distinct from each other and not, sharing the same data structure or something like that.
For me, in C++, basically I use Visual Studio, and it supports so-called OMP. And basically, when I need to parallel something that can be easy paralleled, for example, some for loop, I just write you can write in C++ pragma OMP parallel for num threads and put some number, and that's basically all. So you just write one line and it parallels the loop.
Is it the same as OpenMP?
Yes, it's OpenMP, just short. Basically, I just in last semester, I learned it in the university. So we have parallel programming, so it's this rare, or at least for me, this rare situation when you actually use something you learn in university.
And so the final problem, problem number 5, called Isoform Matching, which is by Andrei Pribelsky, and the tests are by Dmitry Sayutin. There are 2 versions of this problem, an easy and a hard one. It's an interesting formulation of the problem because they spared us, having to perform the alignment. So they already give us the coordinates which have been aligned already, and so there is the sort of the reference genome, and there are the coordinates of the exons and introns. Actually, I don't remember. Matt, can you explain the problem here?
Sure. Yeah, so it's again, they give you, there is a reference genome, right? We don't know what it is, but there is one. And they give you the isoforms along that genome in terms of essentially pairs of start and stop for exons. So there'd be, exon 1 might go from 10 to 20, exon 3 goes from 40 to 400, and there's a bunch in between or whatever. So you have all these isoforms. And then the second thing they give you is a bunch of reads, which are similarly structured in terms of there's a start and stop, and there's a bunch of them in a row. I think the first problem was each read has to match the isoform plus or minus some number of bases. And you have to figure out which one it matches, and if it matches multiple, the total count of how many it matches. And then the hard version was just find the isoform that it most closely matches based on some formula. I think it was like the exon overlap and the intron overlap with some multiplication involved. So yeah, I think that's the gist of it. And it, again, like you said, it matches to the very well-studied, RNA sequencing problems where you sequence the RNA, it doesn't match your reference genome because your reference genome has transcripts that start and stop at particular locations. So they thankfully skipped that alignment step for us. Kind of made it a little bit easier. Just a little bit.
Yeah, so how easy was the easy version?
The easy version I didn't think was too bad. Part of it was I think they didn't quite go crazy on the scale yet. The hard version, they really ramped up the number of transcripts and the length of the genome and the number of reads that you had to process. I don't remember how many it was, but it was a few 100,000, I think, in the largest problem. It took a long time to compute. But the easy one I don't think was too bad. Did you have a different opinion, Max?
Well, basically I don't remember the difference between easy and hard version. So basically, my solution is again quadratic, because for each query, I just look for the best matching. I compare with every isoform we have in the input, and again a little one line of OpenMP and wait for half a hour or maybe a little bit more, and that's all.
So you didn't have to modify your solution at all to accommodate the hard problem?
I modify the function that combines two isoforms because it's different, but the brute force is the same because it's just two loops.
Yeah. I had a similar approach where it was basically brute force with some minor tweaks on whether it's easy or hard. Yeah, I don't know if there's a much better solution there. It was extremely slow in Python, I'll tell you that. It took a lot of parallelizing to get it to finish in time. But I went back to it later just to see how fast I could get it. And I was using Rust, not C++, but you can get it to run pretty fast, even with this kind of like pseudo brute force approach. I just don't know if there's a better way other than just kind of doing a bunch of comparisons and spitting out the correct one at the end.
Yeah, interesting, because for me, the hard problem was too hard. Like for my solution that worked pretty well on the easy version, when I tried it on the hard version, I think it either ran out of memory or it was so slow that I could already see it wouldn't finish in time, like no way. So it is brute force, as you say, but how did you optimize it to be performant enough? Did you use some kind of data structures or anything like that?
Well, first is C++, and second is the function that matches two isoforms. It should be like linear time. So and basically that's all, because it's easy to say that you have to parallel everything, but yes, really you have to do some like this non-asymptotical optimization that make your solution like several times faster.
I do think the people who put this one together were expecting brute force here, because I remember when we were talking to them after the competition was over, they jokingly asked me, how much compute did you have to use for that one? So I think they were expecting some amount of brute forcing of that last problem.
Yes. And I think this problem can be easily make kind of more interesting if you say just don't choose one isoform, but choose like two or several isoforms that better describe somehow come up with your own scoring function. So a group of isoforms that describe new one, and that will make problem like choose k of n, and it will be impossible to brute force.
How would you solve it if it was impossible to brute force?
Well, that's another question. Well, if you
don't tell the competition makers this.
Yes. I have idea one for the problem. Like the full score get like one who gets the more points for the test. So it's like it doesn't matter if your solution bad if all other people have bad solution. And one more thing that for this problem, because at some time I was not sure if it will be enough time, half a hour, or maybe I need 5 hours. So I didn't compare each isoform with each other. I choose 10% of all, and of course this doesn't give full score, but it's like in 10% to the full score. So it's basically if you not competing for the very first place, it's okay to do some very simple planning. It still get high score.
All right, so that's all our problems from the final round. Any concluding thoughts? Like, what are your general impression of the contest?
Yeah, I mean, so I've only, this was the second finals that I've competed in. I'm trying to remember how the first one was I did to see how it compares to that one. liked I felt like a lot of the problems were very connected to real issues this time. Whether that was, just well-defined problems or like the timely who's the super spreader problem, that those kind of things felt very connected to, just the state of bioinformatics. So I thought that was pretty cool. Overall, I enjoy it. Like every time I do it, I have a lot of fun. Extremely tired by the end of it, but have a lot of fun. Yeah, that's pretty much it. I will say I have published my solutions if people are interested in looking at that. So those are on my GitHub, and I'm happy to share that with you somehow.
Oh, cool.
If we can put that somewhere.
That's very nice of you, and I'll put a link in the show notes for this episode.
Sure.
Max, do you want to do the same? Publish your solutions?
Well, I was scared that people will watch it and say that I am right, some shit code, and scared of C++ more, but
my code looks terrifying. Like it's, that code you write on the 23rd hour of the contest is
that's
absolutely gross. Don't use it in production, anyone, but it's fun.
yeah.
Max, anything from you?
Yeah, I also really like contests, especially because I win it, but nonetheless.
Hard not to like it.
Yes. And also I really thankful to the organizers because it may look sometimes like we make a joke from some problems that you just use binary search and that's all. But I was a problem setter a little bit, and I have a lot of friends that are problem setters, and I know how hard it is to come up with problem to make it good for contest, not for just real life or something. And especially when this problem is or when this problem should be related to the bioinformatics, it's much more harder. And they organize it, and in general, it's great, well prepared.
Cool. Was this your first contest, or did you take part before?
Yes, first bioinformatics contest. Well, technically, I have experience with kind of similar problems again, because last time I solved a lot of kind of marathons where you have optimizational problem for a week. And so these problems were quite similar to me. I can say that I have experience solving similar problems, but maybe from other fields. Maybe one more interesting thing. I hope I'm not banned for this, because actually I almost was not alone solving these problems. I remember 1 a.m. I am solving these problems, and I understand that my laptop is not enough to do all the computations. I need one more computer, and I in 1 a.m. I am now stay at dormitory, and I call to my grandmother and ask her to turn on computer and so I can connect to it and run computations on this computer. And it was probably the most hard problem, because I spent 30 minutes to explain how to turn on, how to input password, and how to turn on TeamViewer and everything. Well, that's probably was the hardest problem out of all 8 or all 9 for me.
Yeah. What problem did you need more compute power for?
First, I use parallel programming in first, in fifth, in Superspreaders, so in third problem, and in second I use bin search. So yeah, in 3 problems.
Yeah, I was going to say the fifth one, I think you pretty much had to have like a lot of compute to just get through that one.
Yes.
Yeah, I want to echo shout out to the organizers, because I feel like they do a pretty good job each year putting all this together, and it's always run smoothly for me too. I mean, I know they're using I think it's Stepic, right, for this, but
yes,
it's always very smooth, and I've never had issues just participating in it. So shout out to them for awesome contest again this year.
Thanks, guys, for joining me today, and I hope to see you in the next installments, and I hope to see the next installments of the contest.
Thanks for having us.
Thanks, you too.